For parents to transfer traits to offspring, genes that control each trait must be copied from parent cells and transferred to daughter cells during cell division processes. This week, we are going to study two types of cell division. Cell division is the process by which a parent cell divides into two or more daughter cells. There are several types of cell division depending upon what types of organism is dividing. Organisms have evolved over time to have different and more complex forms of cell division. Most prokaryotes or bacteria use binary fission to divide the cell. In eukaryotes, there are two distinct types of cell division. The first one is mitosis, which is a vegetative division whereby each daughter cell is genetically identical to parent cell. The second one is meiosis, a reproductive cell division whereby the number of chromosomes in the daughter cells is reduced by half to produce haploid gametes. In this session, first you will learn how to prepare wet mouth slide of onion root tip using squash technique that normally employed in cell study. In the real last session, students will use prepared slides to observe mitotic states under microscope. In this online session, we will show you pictures of each stage instead. Before we start, I will explain basic structure of onion root tips. In all kinds of onion, root tips are separated into three different zones, which are zones of division, elongation, and maturation. At the tip of the root, you can see the root cap. The area contains cells that cover and protect the underlying growth region as the root pushes through the soil. Zone of cell division is where the cell mitotic stages occur. Cells are actively dividing but not increasing significantly in size. The cells in zone of elongation is no longer dividing, but their size is increasing. Zone of maturation is where the root cell begins to differentiate into specialized cell types. In this lab, there are several materials equipment to be used. The sample is from the onion root tip. We also use distilled water for washing root sample. Also chemical reagents. There are 1 molar hydrocolloid for softening the cell wall and improve staining. Acetylorcine as coloring agent for staining. We also use other instruments such as microscope slide, cover slip, razor blade for cutting the root tip, tweezer, needle, and brush are used for handling samples. The toilet paper is also used for brothing excess liquid during squash preparation. First, from the onion root tips provided, you can select one using the tweezer to pick the selected sample from the petri dish. Use sharp razor blade to cut the end of the root tip to the length of approximately 2 or 3 millimeters. Please be careful at this step to avoid injuries. Also, please remember that we are going to observe the root tip. Therefore, make sure that you do not discard the tip, but discard the rest of the root sample. In this step, distilled water is very important for cleaning your trimmed sample. First, put the sample on microscope slide. Second, place a drop of distilled water on the sample. Finally, use toilet paper to absorb the water back. We note that the toilet paper can be used in a different ways, depends on individual technique. Then place a drop of 1 molar hydrochloric on the root tip and leave it for 1 to 2 minutes. Hydrochloric will help us to softening root cell wall, as well as improving staining in the next step. After that, wash the sample in distilled water twice. Acetylorcine is used as a staining color. In this step, you will place 2 or 3 drops of acetylorcine on the sample and leave it for 5 minutes. You may use needle to turn your sample around to make sure that it is completely soaked in acetylorcine for better staining. Mount a cover slip on stained root sample. Please avoiding air bubbles. You may add more acetylorcine before mounting for better staining. Squash the slide with your thumb. Please avoid squashing of such force that the cover slip or slide will break. You can blot the excess stain with the toilet paper from the rim of the cover slip as follows step 2. Finally, you can observe sample under microscope. The picture from the right slide illustrates the onion root tip cell under microscope with tenfold magnification. The red color of acetylorcine helps us to reveal the chromosome of sample. You can scan and narrow down to a region containing dividing cells, and also switch to 40-fold magnification for a better view. Before checking the pictures under microscope of mitotic stages from onion root tips, This slide shows interphase and 4 stages of mitosis. Mitosis is divided into 4 stages, except interphase. There are prophase, metaphase, anaphase, and telophase. Interphase is the portion of the cell cycle that the changes is not observable under the microscope. After this slide, you can draw pictures on the lab report that represent mitotic stages of onion root tip. Following the interphase, this stage is called prophase. During prophase, the chromosomes supercoil, condense, and become visible for the first time during the cell cycle. You can see the spindle fibers start forming, and the nuclear membrane starts disintegrating. You have time to observe this slide, and you can pause this video to draw the picture on the lab report that represents prophase stage. After prophase, this stage is called metaphase. During the metaphase, the spindle fibers reach and attach to centromere of each sister chromatid. You can see that the chromosomes align along the center plane of the cell. The nuclear membrane disappears completely. Now you can draw the picture on the lab report that represents the metaphase. The stage after metaphase is anaphase. During anaphase, the centromeres start splitting, and the sister chromatids begin to migrating towards the opposite poles of the cell. We highlight the splitting directions by green arrows. Now we enter the last stage of mitosis. It is telophase. During this stage, the chromosomes are clustered on the either end of the cell. The nuclear membrane starts reforming. The cell plate or new cell wall starts to form between the 2 daughter nuclei. The cell division or cytokinesis will begin at the end of mitosis. We finish watching the online lab of mitotic stages. Now it is time to check details of meiosis, which is another type of cell division in eukaryotes. In the lab report, one chart is presented, then you can check similarities and differences between mitosis and meiosis. Moreover, Don't forget to read the lab menu. See you again for the next online lab.